Guide to the graphic. Here we have a presentation of the liver. Hepatitis A and C. Hepatitis A virus, a picornavirus, with a structure and pattern of replication similar to those of poliovirus, is most commonly acquired from contaminated food and water. After exposure, there is a variable incubation period, 15 to 40 days, during which the virus replicates in liver tissue, but does not cause overt pathology. Shortly before the onset of symptoms, virus is detectable in the blood and feces. As with poliovirus, transmission is by the oral fecal route. Fever, malaise, and nausea are followed by jaundice, a yellow discoloration of the skin and conjunctival tissue caused by the deposition of several bile pigments into the skin and eyes as a result of the liver's failure to remove these products from the blood. The symptoms gradually reverse with time, and a vigorous immune response clears the virus and provides long-term immunity. The incidence of hepatitis A in the U.S. population is about 10 cases per 100,000, and the geographical distribution reflects the fact that poor sanitation favors disease. Many hepatitis A infections occur with no symptoms, and a large percentage of individuals have antibody against this virus. For this reason, injection of antibody pooled from many people, immune serum globulin or gamma globulin, is an effective prophylaxis. Vaccines, both killed and attenuated, continue to be field tested. The hepatitis C virus was only discovered in 1989, although its existence was suspected, and the disease it caused was often termed hepatitis non-A, non-B, to indicate hepatitis not caused by the A or B viruses. A group of scientists from Chiron Corporation, a small genetic engineering company in California, along with the Centers for Disease Control, applied the tools of molecular biology to find this mysterious virus. Starting with a batch of plasma blood proteins that could transmit liver disease to chimpanzees, but that did not cause hepatitis A or B viruses, they concentrated a putative virus and extracted its nucleic acids. They next made a DNA copy of these nucleic acids and placed this putative gene from the serum into a foreign host, the lambda bacteriophage. With correct signals, promoters of transcription, the lambda bacteriophage made a new protein encoded from the DNA copy of the serum virus. Next, antibodies were obtained from people who had had hepatitis transmitted by a transfusion of blood or exposure to blood products, but whose serum contained no antibodies to hepatitis A or B viruses. These patients are classified as having had non-A, non-B disease. Their antibodies were tested to determine whether they would bind to protein made by the lambda bacteriophage, replicating in E. coli and expressing the foreign protein coded by the putative non-A, non-B virus. Remarkably, the research strategy was successful, and a single gene from the new virus, called hepatitis C virus, was eventually isolated and its nucleotide sequence determined. And that is hepatitis A and C. Have a great day.